like to thank everyone for coming today. Appreciate your attendance. And I will talk to you today about a new concept called NutraNanoSpheres and their ability to change the nutraceutical delivery systems. My colleagues, Daniel Strausser, worked with the NutraNanoSpheres. He's in Switzerland. And Dr. Ofumi Akani is, when I talk about the malaria product, you'll see his influence there, the studies we did in Nigeria. The pharmaceutical drug industry suffers from the following. 90% of all pharmaceutical drugs are not water soluble. And 40% of all ages have trouble swallowing pills. And 60% of the people over 60 years of age struggle taking solid medicines. And so many elderly patients can have 20+ pills sitting in their stomach, which diminishes their appetite and causes wasting. 1/3 of the world's population will be elderly by 2025. So bioavailability is a major issue when concerning pharmaceutical drugs and nutraceuticals. And the breakdown of drugs in the stomach can cause dangerous metabolites, cause long-term FDA-approved long-term processes that make approval by the FDA a long process, and the potential for class action lawsuits because of bad pharmaceutical drugs are very prevalent. And biologic drugs require the intravenous injections. They cannot survive the stomach. So these are some of the pharmaceutical problems we have. And it's difficult to modify dosages. And as we'll see, we'll show you how this can be done quite easily with the NutraNanoSpheres. So today I'm going to introduce a new type of pharmacology called NutraNanoSpheres, or NNS. You'll see some of the nomenclature of that. And basically, what we have is we take a fatty acid, and by the laws of thermodynamics, the water-fearing regions, these carbon-hydrogen groups will congregate inside, and they will replace externally the carboxylic acid charge groups on the outside. And they'll spontaneously form these beautiful spherical spheres. And what we do in this formation, we will introduce a nutraceutical, for example, curcumin, when we make the process of making the NutraNanoSpheres, and it will trap the curcumin inside the NutraNanoSphere. Now, what happens, we have proprietary methods, which I won't go into today, of doing this encapsulation. Enables us to, we've encapsulated over 300 different compounds with this process. And so these micelles that form around whatever nutraceutical product we're interested in, we just mix them with it, and we cause this formation. And it doesn't require just a slight amount of heat. It does not use organic solvents to do this process. It's all natural in the way we do it. So this process enables us to have high bioavailability and potency for our nutraceuticals. And I'll show you some examples of the applications of that. Now, a very important thing when you're making a micelle and you're using it, micelles are only stable if you make them small enough. Micelles that are above 100 nanometers do not have a long service in existence. The Malvern Instruments, who makes the instrument that we use to make these measurements, I'll go into that in a moment, have noted this over the years, and this is actually their graph. If you're dealing with 100 to 300 nanometers, you're talking about stability in days or months. But when you go out to our NutraNanoSphere range down to 3 nanometers to 10 nanometers up to 30, we can our products are stable for years, and a lot of them at room temperature. And our NutraNanoSpheres are so stable that you can heat them to 98 degrees centigrade, and they won't deteriorate. Not saying what's inside will deteriorate, but that's important too. But the advantage there is that we can use it as a hot fill for the soft drink industry, because they require 178 degrees for 2 minutes in their filling process. So that's the importance of that. Now, I'm about to show you some graphs using where we measure the average diameters of the NutraNanoSpheres and also measure what we call polydispersity. And that is how broad are the peaks that we measure. So we take dilute samples, we filter 0.22 microns, and put them in a Malvern instrument, or also we use the Brookhaven instruments. And using a non-negative least squares algorithm, we can generate size distributions and also the distributions, not only the diameter of the particle, but also the dispersity of it, the polydispersity we call it, of these highly bioavailable NNS. NNS is NutraNanoSphere. I may say that acronym. Now let's look at the industry standards in making measurements with our NutraNanoSpheres and making particle measurements. The instrument standards by the National Institute of Standards and Technology in this case, we use a 31 nanometer traceable standard and also a 101 nanometer traceable standard. These are made actually from silica, from glass. And I won't go into detail how those are made, but these are the standard curves that you get. Now, 2 important things here to look at is the effective diameter and also the polydispersity, and also the reproducibility of running these. But here we have the size of this 31 nanometer traceable standard. It has a polydispersity of 0.04. Smaller that number, the sharper the peak, better the preparation. Here is a 101 traceable standard, and you see it has a polydispersity of 0.005, extremely good. So these are good internal standards we use when we make our measurements. Now, let's look at some of the biologic products that we have. One of them is curcumin. I'll show you the importance of curcumin. Again, we have many products that we've made, and I actually brought some here to show you later on if you're interested. Here's 2 separate preparations of curcumin that's incorporated into our NutraNanoSpheres, and you see the polydispersity is 0.016 and 0.011. Now in this particular preparation, we call it triple curcumin because we use all 3 of the natural occurring curcumins. So all of our preparations, we try to stay as near natural as possible. even though curcumin itself is 80% of that population. Now, if we compare the 31 nanometer standard, here it is at 0.04. Here we got 0.01 with our curcumin. It's better than the standard that's used to align the instrument, to set up the instrument. Here's another example. Here's the standard at 101, and here is vitamin D3, which actually is a very large diameter. We can make vitamin D at 7 nanometers, no problem. European FDA requires us to make it at 142. And there's a reason for that. We would highly compete with the drug industry in Europe if we were not allowed to do that, but we can do it in the US. But that's all I'm showing you here. Here's the polydispersity, very, very good, at 142 nanometers. Now, here are some of the products, just a few of them that we make. Usually we make them in a 15 ml container with a very narrow nozzle. So 20 drops is a half an ml. And so we normally recommend 20 drops, and it's a 15 ml bottle. So you got enough for a 30-day supply. So that's how we present it. We're now presenting in a 30 ml bottle where you take 1 ml, but you take mixtures of all of these, so you don't have to count drops all the time. So anyway, these are all very useful nutraceuticals. The one here in the middle is called Tri-Anti-VP, and I'll go into that in a moment when we talk about the malaria product. But I will mention that the Tri-Anti-VP is also very antiviral, and we used a lot of it, at least 70 or 80 patients during the COVID episode to get them out of the hospital, off the respirators. Very effective as an antiviral. Now, we'll look in closing, we're going to look at a few examples of the application of the NNS, NutraNanoSpheres, which apply to the clinical environment. The first one is a number of years ago, my wife and I, who's now sitting here in the audience, Benita, we've done medical missions in Haiti for over 10 years. And one year I went in May, they delayed it because of security issues. It's very frequent in Haiti. First time I saw malaria. I knew what malaria was. And I saw these children suffering from malaria, and we took care of helped a home of 34 children. And I'll show you the lady, Roberta, who we congregated with a lot during that time. That children, if we treat, and I don't have time to go into details of where I arrived at this. If we treat with 50 milligrams of artemisinin, 50 milligrams of curcumin, and both of these, artemisinin is 99% pure. The curcumin we use is 97%, and 50% citrus bioflavonoids. We made green gel caps that we were able to treat for malaria. And the proposal that I used when I looked at this, I said, if we're going to treat children for malaria, when all the other conventional treatments only last long enough till the next mosquito bite, which is probably in 30 days. So what I did, being partly an immunologist, I'm PhDs in chemistry and biophysics, but I love immunology. So I decided to treat over 16 days. It's not uncommon, with antibiotics and things to be taken 14 days in Haiti, for example. And we treated these patients every day with In adults, 2 of these green gel caps, and the children we treated with 1 below the age of 12. And what happened was the malaria disappeared within 5 to 7 days. It didn't go down as fast as your conventional chemotherapy drugs, but when it went down, what we were doing was breaking up the parasites with these compounds and exposing them to the immune system. And I've shown and published this with Buffy Coates with work I've done in collaboration with Harvard University, where we showed that the amplification studies of the Buffy coats from these children were able to measure DNA amplification and show that the humoral, the cell-mediated immunity, and even the defensins that are produced are all elevated in these children after our quote-unquote in vivo immunization process. And as you can see here, we measure mean parasite load. And basically what that is, and it's a WHO-approved method of looking through a microscope and counting the number of parasites that are in the red cells in a certain area, and you'll see them disappear. They go down to 0. And here we have children that we cured 90% of them. 100% of the adults, 100% of the adults and children in Haiti. What I'm showing here is data from Nigeria. And so with that excitement of seeing those results, then we started looking at babies because they're the most vulnerable group to being treated, because you can't give them tablets or gel caps. So we went back and took our NutraNanoSpheres, curcumin, artemisinin, and then we used bilberry as our antioxidant involved in this. And those 3 things is in that tri-anti-VP I showed you. We gave that to babies in baby's milk at half the dose of the children, so 25 milligrams of each. And in 16 days, we cured 93% of the babies. Now, did it last? We went out 60 days. We've gone out 3 years with children. But here's an example of where we treated. We had 1 recurrence at 30 days. And this is 15 children. We did another, and you see the mean parasite. This is just showing you the table, showing going, all of the parasite disappeared, except for this 1 case. Now, here are a group of children, and this is Roberta in Haiti. I treated Roberta 6 years earlier, and this is a 2015 photograph, so you got to add the years onto that. They didn't ever got malaria again. And you can see the ages that we treated and what age they were at that stage. We've actually had 2 of our children go to our local university. 1 became a nurse and 1 became a theology professor. They never got malaria again. In fact, the fellow who went to our university for the theology degree, his daughter was on her deathbed, and the preacher came to administer last rites to her, and we got her to take this medicine in 3 days. She was up, and she's never got malaria again after the 16-day treatment. The only negative thing about our treatment, at 3 days you start feeling pretty good. But we tell them it's like going to dip in the River Jordan 7 times. You got to take this medicine 16 straight days. You take it 8 days, it doesn't work. Okay. So all of that is an introduction to what I'm about to show you, what we've done for the babies. These babies is as young as 2 weeks of age, we treated with malaria. They had ant bites. Umbilical cord was still in the can. When somebody dropped a baby off at the orphanage. Now they're healthy adults. So that's one example of our application. Another example, and I've got a lot I can show you, is the one I picked before this meeting, and that's using choline. Now, if you're not familiar with choline, choline is, first of all, it's an essential nutrient. Your body can't make it. The liver can make some of it, and but not very much. But it's the precursor to acetylcholine, which is the neurotransmitter, and also an S-adenosine methionine universal methyl donor. But the importance here is choline actually helps you produce new neurons in the process. Now, why is that important? Well, if you're looking, 44% of pregnant women are deficient in choline. That means the child brain development is impaired. And there have been published studies showing taking choline all the way through pregnancy and even 5 years old, that those children's cognitive abilities are much better than children not taking it. So, and also, like all our products, we can cross the blood-brain barrier with our NutraNanoSphere because they're intact. When they leave into the bloodstream, they're intact. And but you need 14 micromoles of choline to cross the blood-brain barrier. That's the critical concentration. In our system, using 10% choline, it turns out to be 100 micrograms per 1 ml. We use 1 ml pipetters. We can get 1,601 micromoles per liter. Huge increase. So we think this is going to make a huge effect on childhood development. And now, the last illustration I'll show you here of an example of what we use, we can take vitamin C and we can incorporate it into a NutraNanoSphere as small as 3 nanometers. That'll go right through your skin, by the way. And if we take the 3 nanometer one and we put another coat on it, and then we take a third NutraNanoSphere and make a third coat, then we mix these various groups together, we have an 8-hour release vitamin C. So that's one of the applications we can use. Now, what's so important in what I'm presenting to you is the very last part here, and that is you can conceive that the NutraNanoSphere will go through the stomach, go through the gut, into the bloodstream intact, and that NutraNanoSphere can then go into a cell, and we have shown that we have shown with vitamin C, for example, if you take vitamin C and you put it in culture with a K562 tumor cell line, it has absolutely no effect up to 10,000 micromole. Vitamin C does not kill the cancer cell. If you take the vitamin C in the NutraNanoSphere, it will enter that cancer cell and kill at 125 to 140 micromole. And that's the LD50, the lethal dose 50%. And by the way, we use an isotonic propydium iodide to do the cell viability assay. it will passively stain live cells, but the fluorescence goes through the roof when it binds to the DNA in the cell. So that's how we do that assay, by flow cytometry. So that's the importance of the NutraNanoSphere, being able to penetrate whatever cell it's going to have action on. Okay. Another example, if we take curcumin and we solubilize it with DMSO. So what we're doing is taking something that's insoluble to try to make it soluble enough that we could do a cell culture with, and the DMSO has no effect on the tumor at that concentration. And what we see is that the curcumin can enter and is cytotoxic to the cancer cell. And the LD50 for that is 11,000 nanomoles per well. And when I say per well, we have 100,000 cells that we culture during this period of time. We can measure. If we use NutraNanoSphere curcumin, the LD50, the lethal dose is 34 nanometers per well. It's 324 times better at killing the cancer cell than if it was just free curcumin. And here you can see the X's are the 50% killing, and we can see that quite easily in our measurements. Now, just one more note about curcumin, and is that when we add the curcumin, the NNS, the NutraNanoSphere curcumin, to a cell culture, it kills it immediately. And there's a whole bunch of mechanisms. I could sit here and list them for you for the function of curcumin. It's very, very important. So it can kill in this 30-second exposure, and that's the time it takes to mix the propylium iodide stain and put it on the flow cytometer. It happens, I think, spontaneously in killing cancer cells. But it can enter normal cells and does nothing but provide antioxidant and all kinds of other activities that are beneficial to normal cells. Now, finally, the characteristics of the NutraNanoSpheres. I'll just summarize those here. We can make extremely small sizes. They're very important because it makes the cells, makes the NutraNanoSphere stable. 3 to 10 nanometers. We usually go up around 28, or depends on if we mix different groups together. I brought some samples today, like I'll show you. I've got a brain energy formulation, has 18 components in it, all NutraNanoSpheres, all you take with 1 ml. The stability is measured in years. It is stable even at 98 degrees centigrade. And the preparations that we use do not use organic solvents or high temperatures. And we're talking about moderate temperatures in the 50-degree range. So we certainly can use it with proteins and peptides. Preparations can be made in hundreds of liters. We have generators, reactors to do that. And it's biostable for polypeptides. It can survive the stomach and provide the intact NutraNanoSpheres to the bloodstream. The bioavailability is important. The accessibility is even more important. And no one really addresses that except us. And more importantly, it needs to penetrate cells intact. And also it's a very economical product to produce in our environment. Now, that's the summary, but that's not the end. I want to show you. Can you do that? Sure. a little animation for you that my son Kyle did. He's a computer animator. And forgive me for showing you a liposome at the beginning, but that was back in the day. Turn that sound.
NutraNanoSpheres are water soluble.
Okay. You can turn it up a little bit, I guess, the narrator. Don't pay attention to that. We're dealing with micelles, not liposomes. Okay. But I just want to show you just the structure of it, and you can just watch this on your own. And see, we can drop it. You drop these NutraNanoSpheres in water, and you don't even have to stir it. They just go right into solution and go through go right into your bloodstream. And then travel intact to attack whatever cells that are react with whatever cells you're interested in. I will tell you in passing, we've developed a glutathione treatment for this, a 10% glutathione. You don't have to take IVs anymore. Works just as well. Okay, that's it. Escape.